We're going to take a look at how cells divide. So firstly, we're going to talk about how prokaryotic cells divide. So cells like a bacteria or single-celled organisms. So the way in which they divide is through binary fission, which basically means to divide in half. And that's what prokaryotic cells like a bacteria are going to do. Prokaryotic cell will divide into two, divide in half, divide into two identical cells. So we start off with a simple prokaryotic cell like so, and in our prokaryotic cell we would have a single looped chromosome like so, double-stranded chromosome, which contains all of the genes, all the genetic information required in order to make a cell or make a copy. Now, the first thing that's going to happen, if we go to sort of step 2 of binary fission, is inside this prokaryotic cell, we will have DNA replication take place. So now, instead of having one single loop of DNA, we have two. So if this cell is going to divide into two, divide in half, and create two new identical daughter cells, we need an identical copy of DNA. So that's what we end up with. We have DNA replication take place so that there's now two identical copies of that single looped chromosome inside this prokaryotic cell. Now, what will occur is each of these single loop chromosomes will, I'll try and make it relatively obvious, attach itself to the membrane. So they actually connect to the membrane like so. And this is because what we'll see occur is the cell will start to grow outwards. Now, as the cell grows outwards, it's going to pull apart and take with it the copy it has of the replicated DNA. So as we see the cell start to grow outwards, it will take away its copy of the DNA, which is required in order to become a new cell and to be able to direct creation of different proteins and cellular processes and things like that must occur in this prokaryotic cell. What you notice also here is that the cell starts to pinch inwards, and this is the early stage of the cell being split or divided into two. And finally, once this pinching has taken place in its entirety, we will now end up with two new prokaryotic cells. This loop of DNA will detach from the membrane, and we now have two identical daughter cells, which are exact copies or clones of the original. Now, this process can happen quite rapidly because it's so simple. It's able to happen quite rapidly, quite quickly, and under desirable conditions, what we'll see is an exponential rate of growth. So over the course of time, what we will see is that because one cell is dividing into two, two into four, four into eight, etc., if conditions are good, binary fission is able to occur exponentially. So bacteria or the like prokaryotic type organisms are able to divide very rapidly, which makes them really good for testing and things in lab environments, because division happens really quickly. We can see results really quickly as well. So that is binary fission.